Welcome to the Deep Dive. We're the show that cuts through the noise to get you the key insights from new research. And today, wow, we're tackling something really fundamental in cancer, how it actually progresses. Because, just knowing how big a tumor is or if it's spread, well, that doesn't give you the whole story, does it? It doesn't tell you what's happening inside. That's where this idea, the hallmarks of cancer, comes into play, these basic abilities that cancer cells pick up to, well, thrive and take over. But here's the snag, and this is what we're really digging into today. We have this great framework, these hallmarks, but our everyday diagnostic tools, they rarely measure them directly. And that leaves this huge gap in understanding how a specific tumor is behaving, which makes tailoring treatments, well, much harder than it maybe needs to be.
Exactly. You've hit the nail on the head. Think about the standard ways we stage cancer, like AJCC or T&M staging. They're good at describing the physical stuff size, lymph nodes, metastasis. They tell us where it is macroscopically, but they often just glide over the molecular changes, the stuff that's really making that tumor tick. And that's why, you can get two patients, same stage on paper, but then boom, completely different outcomes, because molecularly, their cancers are just running different programs.
Okay, so that sets up our mission for this deep dive. We're going to explore a really exciting new framework called OncoMark. It's been developed by researchers, and the idea is to bridge that gap we talked about. It aims to estimate the activity of all 10 cancer hallmarks simultaneously using gene expression data, the big picture goal, to get this detailed molecular snapshot of a tumor, hopefully leading to much more individualized, much more effective treatments. All right, let's back up a second. These hallmarks of cancer, what exactly are they? You hear the term, but what does it mean? We're talking about work by Hanahan and Weinberg, right? The foundational stuff.
Absolutely. Their work was, transformative. It gave us this kind of roadmap for how cancer cells evolve, the core capabilities they have to acquire. Initially, I think it was 6 hallmarks, and then later, as we learn more, it expanded. Now we generally talk about 10, plus maybe a couple of enabling characteristics.
10 core abilities. Okay, can you break those down for us? What are we actually looking at?
Yeah, sure. Let's run through them quickly. First, sustaining proliferative signaling. Think of it like the gas pedal is just jammed down.
Right.
Uncontrolled growth, non-stop dividing.
Okay, number 1, grow, grow, grow.
Number 2, evading growth suppressors. So the body has brakes, right? Cancer cells figure out how to cut the brake lines. They ignore the stop signal.
Got it. Ignore the brakes.
3, resisting cell death or apoptosis. Normal cells, if they're damaged, they self-destruct. Cancer cells, they learn to tough it out, survive things that should kill them.
So they're hard to kill.
Very. 4, enabling replicative immortality. Most normal cells can only divide a certain number of times. Cancer cells? They basically find the fountain of youth. They can just keep dividing forever. Limitless replication.
Immortality, wow. Okay.
5, inducing angiogenesis. Tumors need fuel, so they trick the body into building new blood vessels directly to them, ensuring they get nutrients and oxygen.
Like building their own supply lines.
Exactly. 6, activating invasion and metastasis. This is the scary one for many people. The ability to break free, travel, and set up shop in distant parts of the body, colonizing new territories.
Spreading. Okay, what else?
7, deregulating cellular energetics. They change their metabolism. It's like they switch to a high-octane fuel to support all that rapid growth.
Right, changing their energy use.
8, avoiding immune destruction. Our immune system is supposed to hunt down and kill rogue cells. Cancer cells develop ways to hide, like putting on camouflage, or actively fight back against immune cells.
So they evade the police, essentially.
Pretty much, yeah. 9, genome instability and mutation. This is more of an enabler. They become really unstable genetically, making lots of errors when they copy their DNA. This speeds up their evolution, helps them find new ways to survive.
So they adapt faster.
Much faster. And finally, 10, tumor-promoting inflammation, another enabler. They can sort of co-opt inflammation, which is normally a healing response, and turn it into something that actually helps them grow and spread, creates a supportive neighborhood for the tumor.
Wow. Okay, that is a complex and, frankly, terrifying set of skills.
Yeah.
And yet, you said earlier, we don't routinely measure these in the clinic. Despite knowing all this, why the disconnect?
Well, it comes back to the tools. Traditional methods just aren't designed to capture these dynamic molecular changes happening inside the cells. They give us a static picture, mostly anatomical, to really get at personalized treatment, to know why a tumor is aggressive or what its specific weak spots might be. You really need to understand these hallmarks. It's kind of the foundation for precision oncology.
Okay, so if the old tools aren't cutting it for this molecular view, what are researchers doing? What's the cutting edge? That brings us back to OncoMark, right?
Exactly. OncoMark is presented as, and this is a big claim, the first computational tool specifically designed to predict all cancer hallmarks concurrently, at the same time. That's the key innovation here.
All 10 at once. How does it do that?
At its core, it uses something called a neural multitask learning framework. Fancy name, but basically it takes transcriptomic data that's the readout of all the gene activity from a tumor biopsy.
Okay, like a list of which genes are switched on or off.
Pretty much, yeah. It takes that complex data, feeds it into this AI model, and the model then calculates the probability, the likelihood, that each of those 10 hallmarks is active in that specific tumor. So you end up with this detailed molecular profile, sort of hallmark activity score.
That sounds incredibly powerful, like getting under the hood of the cancer cell.
Oh, man.
But hang on, how do you train something like that? You said yourself, we don't have like a big database of tumors already neatly labeled with hallmark X act, hallmark Y inactive. There's no experimental gold standard, as the researchers put it.
Ah, yes, that is the million-dollar question, isn't it? And their solution was really quite ingenious. They couldn't use real biopsies for initial training because they weren't labeled, so they created synthetic data.
Synthetic. Like fake biopsies.
Sort of. They started with real data, but at the single-cell level. Massive amounts. 3.1 million individual cell blueprints, their transcriptomes, from nearly 1,000 patients across 14 different cancer types.
Okay, so they have the raw ingredients.
Right. Then, they carefully figured out which sets of genes are linked to each hallmark, being really strict, only using gene sets confirmed by multiple studies. They used computational methods to score individual cells for hallmark activity based on those gene sets. Then, and this is clever, they turned those scores into simple binary labels. Hallmark positive or hallmark negative.
On or off, basically.
Essentially, yes. But they did this carefully, adjusting the thresholds for each tissue type because, say, lung cancer might express genes differently than breast cancer even for the same hallmark.
Okay, that makes sense.
And then they digitally combined cells that had the exact same hallmark labels to create these synthetic pseudo-bulk samples. These mimic real biopsy data, but crucially, for training, they focused on creating samples where only one hallmark was labeled as on.
Why only one? Real tumors have lots going on at once, right?
Exactly. Real tumors are complex mixes. But for training a supervised model like this, starting with clean, unambiguous signals, these non-overlapping hallmark labels, was critical. It ensures the model first learns to recognize each hallmark signature accurately, without the noise of coactivation. It was a deliberate design choice for clean training.
Okay, I see. Start simple, build complexity later. But that leads to the next big question. If it's trained on these clean, one hallmark at a time synthetic samples, how does it possibly cope with the messy reality of real tumors, where multiple hallmarks are often firing simultaneously and interacting?
That is precisely where the multitask learning or MTL architecture comes in. It's not just 10 separate models. It has shared layers, a kind of central processing unit.
A shared brain.
Yeah, a shared brain, that's a good way to put it. This brain learns general patterns and features relevant across all the hallmarks. Then it has separate output heads for each specific hallmark. This structure allows it to leverage those shared biological signals it learns and figure out the coordination patterns, the crosstalk between hallmarks, implicitly. Even though it wasn't explicitly trained on coactivation, the shared learning allows it to generalize. It bridges the gap.
Okay, that's quite sophisticated. It learns the basics cleanly, then figures out the complex interactions. So the big test, how well does it actually work? How did they validate it?
The validation results are frankly stunning. First, they did internal cross-validation, testing it on parts of their synthetic dataset it hadn't seen during training, and the scores were incredibly high. Accuracy, precision, recall, F1 scores, all consistently exceeding 99%.
99%. Wow.
Yeah, for some hallmarks, like sustaining proliferative signaling, it hit 100% across the board. Even for complex ones like activating invasion and metastasis, it was like 99.97% accuracy. Really impressive.
Okay, that's on the synthetic data it was kind of built from. What about completely independent real-world data?
That's the crucial test, right? External validation. They tested it on 5 completely separate independent data sets from other research groups, data it had never seen before, and it held up remarkably well. It consistently maintained a minimum of 96.6% across the key metrics. In some cases, on certain external datasets, it achieved perfect metrics with 100% accuracy.
That's very strong validation.
It is, and there's more. They specifically tested if it could tell the difference between normal tissue and cancerous tissue based on hallmark activity. In cancer samples, OncoMark showed significantly higher probabilities for these hallmark signatures being active. In normal tissues, the patterns were, as you'd expect, tightly controlled, mostly inactive. They even used statistical tests like the Kolmogorov-Smirnov test, which confirmed these differences were highly significant, with p-values effectively 0. It clearly learned meaningful biological patterns, unlike standard machine learning models they compared it against, which apparently just defaulted to predicting inactivity.
So it seems robustly validated, both internally and externally, and it can distinguish cancer from normal tissue based on these hallmarks.
Let's bring it back to the clinic. What does this actually mean for patients? What are the real-world implications here?
This is where it gets really exciting. For instance, remember hallmark number 6, activating invasion and metastasis?
The spreading one, yeah.
Well, OncoMark predicted elevated activity for this hallmark, even in the primary tumor samples. The original tumor site of patients who were already known to have metastatic disease elsewhere.
So it could see the spreading potential even in the main tumor.
That's what it suggests. It seems to capture these early biological signatures, this metastatic competence, potentially identifying tumors that are poised to spread or have already started seeding micrometastases. That could be huge for treatment decisions.
Definitely. What else? They also looked at how hallmark activity changes with traditional clinical cancer stage, and they found this clear, dynamic progression. Generally, hallmark activities ramped up from stage I through to stage 4. The most complex co-activation, multiple hallmarks firing together, was seen in the most advanced stages.
So it mirrors disease progression, but on a molecular level.
Exactly. It highlights how these hallmarks drive the increasing aggressiveness of the cancer as it advances. It's like watching the tumor accumulate these dangerous capabilities over time.
Okay, that makes sense biologically, but the real payoff would be linking this to treatment, right?
Absolutely. And this is probably the most practical implication. OncoMark appears capable of linking specific hallmark activity profiles to how patients respond to certain drugs and even to their overall survival outcomes.
Oh, so give us an example.
Okay, so they analyzed patient data where they knew the treatments received and the outcomes, like how long they were disease-free or how long they survived overall, and they found really strong connections. For example, if a tumor showed high activity for enabling replicative immortality, that limitless division we talked about.
Right, the immortal cells.
Patients with that profile seem to benefit more from drugs like anastrozole, particularly regarding disease-free survival, or if resisting cell death was highly active, drugs like trastuzumab or vinorelbine seemed more relevant, especially for progression-free survival.
So you can match the drug to the specific trick the cancer is using.
That's the idea. It pinpoints the tumor's specific vulnerabilities based on its hallmark profile. The data showed numerous links like tumor-promoting inflammation activity being linked to response to radiation or inducing angiogenesis linked to irinotecan for overall survival. It allows you to say, okay, this tumor is heavily reliant on building blood vessels, angiogenesis. Let's hit it with an anti-angiogenic therapy. It's moving towards truly targeted therapy based on the tumor's functional wiring diagram.
That really does sound like the future precision medicine guided by these functional hallmarks. It's incredibly promising. But let's be realistic. What are the hurdles? What needs to happen before you or I could walk into a clinic and get an OncoMark profile?
Yeah, there are definitely challenges. Like any advanced tech, especially involving genomics, there are practical hurdles. First off, the high cost of transcriptomic profiling, getting that initial gene expression data, is still a factor, though costs are coming down.
Okay, cost. What else?
You need specialized infrastructure labs and computational resources to run the analysis. And then there's the big one, integration into clinical workflows. How do doctors actually use this information easily alongside everything else? That requires changes to systems and training.
And you mentioned earlier the lack of ground truth data in real biopsies for perfect validation outside the synthetic setup.
Exactly. While the validation is strong, ongoing real-world clinical studies are crucial to confirm its utility across diverse patient populations and truly benchmark it against patient outcomes prospectively. We need more data from routine clinical practice.
So, significant hurdles, but are the researchers optimistic? Does the field see a path forward?
Oh, definitely. There's a lot of optimism. As I said, the declining cost of sequencing is a huge driver. Plus, we're seeing the development of portable sequencing devices. Imagine getting this kind of data faster, maybe even closer to the patient. And cloud-based analytical platforms are making the heavy computation more accessible. You might not need a supercomputer in every hospital.
Right, making the tech easier to access.
And we're seeing better integration with electronic health records, which is key for making this information usable for doctors in their day-to-day work. So the pieces are starting to fall into place.
Well, this has been an absolutely fascinating deep dive. We've explored OncoMark, potentially the first computational tool to predict all 10 hallmarks of cancer simultaneously. The potential seems enormous. Better prognosis, much sharper therapeutic targeting, really pushing forward that dream of precision oncology.
It really is exciting, and it makes you think. Like, considering how dynamic these hallmarks are, and if OncoMark can potentially track them, what does this mean for understanding how cancer evolves within a patient? Or maybe even more profoundly, could tracking these hallmarks in real time, perhaps even from something like a blood test down the line, totally shift our focus? Could it move us towards preventing the full set of hallmarks from ever establishing rather than just treating them once they're active?
That's a powerful thought to end on, moving from treatment to proactive prevention based on this molecular understanding.
Yeah.
Plenty for all of us to think about. Thank you for joining us on the deep dive today. We hope you continue exploring this incredible intersection of cancer research, technology, and the future of medicine.